Reverse cholesterol transport. Transport of cholesterol from extrahepatic tissues to liver for disposal. Unlike other lipids, triglycerides or phospholipids, cholesterol cannot be degraded in our body. It has got this large steroid nucleus, which is our body is not able to degrade. So cholesterol is taken to the liver from where it is disposed of by the hepatobiliary system as bile salt or as neutral sterols. Cholesterol, a very useful compound. You can see here precursor for a large number of compounds, vitamins, hormones, bile acid, bile salt, membrane component. But excess of cholesterol is bad because it can lead to CAD, coronary artery disease, brain stroke, the biggest killer disease of the world today. That is why cholesterol is portrayed as a villain, but it is not only thing, its excess is bad. And today we are going to see how it is eliminated from the body, a function performed by HDL or the high density lipoprotein. So the high density lipoprotein is responsible for carrying cholesterol from extrahepatic tissue to the liver, from where it will be disposed of, eliminated from the body. Some other lipoproteins also, VLDL, LDL, etc., they also participate in reverse cholesterol transport, although in an indirect way. We are going to talk, although their main function is to carry lipids from liver to the extrahepatic tissue, but how they indirectly participate in cholesterol disposal, we are going to see again here. But the principal player is HDL, high density lipoprotein, high density, higher than any other protein component is highest in it. And the size also is lower. Also called alpha lipoprotein because it forms alpha band in the electrophoresis carried out at pH 8.6. So now we come to reverse cholesterol transport. The main actor, as we said, is HDL. The various stages of metabolism are nascent HDL, HDL2, HDL3, etc. First of all, nascent HDL comes into picture. This is the first one which is produced from intestine and liver. और अब हम देखेंगे that this is going to carry cholesterol from peripheral tissues and take it to the liver from where it will be eliminated. But usse pehle, it acquires other lipoproteins from the other proteins, apolipoproteins from the other lipoprotein fraction. In nascent HDL, it is nothing but it's a phospholipid bilayer with E1, apoprotein E1 at the edge. So after it obtains other lipoproteins, after it obtains cholesterol with the help of a protein called ABCA1, ATP binding कैसेड प्रोटीन ए, तो ये जैसे इसके अंदर कोलेस्ट्रॉल आएगा, so it will be converted to the mature form of HDL. So nascent HDL acquires other protein fractions from other lipoproteins. It also acquires cholesterol with the help of this protein ABCA1 and gets converted into mature HDL. Mature HDL, now comes action of two enzymes, LCAT and CETP. LCAT, lecithin cholesterol acyl transferase, and CETP, cholesterol ester transfer protein. तो ये HDL 3 फॉर्मेशन हो गया. तो इसके सरफेस के ऊपर these two enzymes are there. So what actions do they perform? Let us going to see. LCAT converts cholesterol into cholesterol ester. ये जो कोलेस्ट्रॉल जो पेरिफेरल सेल से आया था HDL के अंदर, it is converted to cholesterol ester by LCAT, lecithin cholesterol acyl transferase. Lecithin, all of you know, it is a phospholipid containing two fatty acids, and then there is a head group. So this fatty acid is transferred to cholesterol to form cholesterol ester. As you know, at the third position of cholesterol, there is a free OH group. So that OH group के अंदर अगर ये fatty acid attach हो जाए, so this will form cholesterol ester. So what does it, what change it induces in the HDL? Cholesterol ester being completely hydrophobic compared to free cholesterol, which is amphipathic. This is completely hydrophobic, so it sinks to interior of the HDL particle. As a result, the HDL becomes more and more spherical. It heads towards maturity. It becomes mature HDL particle. Then what happens? Mature HDL, it is rich in cholesterol ester. This second enzyme CETP, cholesterol ester transfer protein, as the name suggests, it transfers cholesterol ester to the VLDL, very low density lipoprotein, in exchange for triacylglycerol. So ये HDL3, it loses its cholesterol ester to VLDL, and in exchange it acquires TAG. So this was rich in cholesterol ester. Now this is poor in cholesterol ester. Now it is called HDL2. So this is one point where undergraduates, they make a lot of, lot of confusion is there. So this point I have tried to clarify. One more point here. Now, HDL2, it is acted upon by an enzyme called hepatic lipase. I think I have shown it here. It enters the space of DC where it is acted upon by an enzyme called hepatic lipase. Hepatic lipase causes degradation of phospholipids and triglycerides. And while this degradation is occurring in the space of DC, the cholesterol is taken up by the liver cells by certain specific receptors. We are not going to talk about those details now. So this is the first route of reverse cholesterol transport mediated by hepatic lipase. Some HDL particles, they acquire ApoE. A minority of HDL2 particles, they acquire ApoE, apolipoprotein E, or apolipoprotein E containing surface के ऊपर these bigger HDL particle, they will be taken up directly into the liver cell. They are endocytosed. So this is the second route of reverse cholesterol transport. पहला HDL hepatic lipase mediated. Second is ApoE. Note the difference between them. यहाँ पर full HDL particle is endocytosed. Here full HDL particle is not endocytosed. Only the cholesterol is transferred to the into the liver cell. Just a bit of digression I'm entertaining. You have seen here that apolipoprotein E, it is acting as a ligand for the receptor so that the HDL can be taken. तो एपोलाइपोप्रोटीन का एक मेजर फंक्शन है एक्टिंग एज लाइगैंड. सेकंड पॉइंट, A1 is stimulator of LCAT. So this is second important role of apolipoprotein. They stimulate the enzyme activity. More examples you can find out yourself. So अभी तक हमने दो एग्जाम्पल, दो रूट्स देख लिए. बोथ आर, यू कैन क्लासीफाई एज डायरेक्ट रूट, एंडोसाइटोसिस ऑफ लार्ज एपोई containing HDL particle, और दूसरा हेपेटिक लाइपेज mediated in the space of disease. Third route is indirect route. What is that? हमने ये देखा था, कोलेस्ट्रॉल ईस्टर VLDL में चले गए. I have written VLDL remnant in that slide. We will be talking about that in some other lecture when we discussed about VLDL degradation by lipoprotein lipase. So this VLDL, it will be converted to remnant particle, then LDL, and as you know, LDL is taken up by the liver. So this forms an indirect route. HDL से कोलेस्ट्रॉल ईस्टर गया VLDL में, और VLDL IDL LDL एक्सिस से हेपेटोबिलियरी एक्सक्रीशन हो गया. So this is the indirect route. So the three routes we have discussed, two are direct, one is indirect. An important point, indirect route, the indirect transfer through the CETP and remnant particles is quantitatively the most important in humans. Now, these are some major points. तो आपको इसको खुद रिवाइज करना है अपने आप. Why this is called ABC protein बोलती है, why it is called scavenger protein, why LCAT, CETP are important, और इनकी deficiency में क्या होगा, that also you should try to see. Hepatic lipase का क्या role है, और what is HDL3, HDL2. Now a clinical correlate towards the end. The premenopausal women have lower risk of CAT. The answer is very simple, because under hormonal influences, I think estrogen होगा, है ना, estrogen, इनका HDL level high है males के मुकाबले. And HDL, as we have seen, it is a scavenger of cholesterol. It drains cholesterol from the cells. तो सेल से ड्रेन करके लिवर से एक्सक्रीट कर देता है. तो इसलिए HDL is also called as good cholesterol. A clinical problem, दो पेशेंट, दोनों का टोटल कोलेस्ट्रॉल इज सेम, बट HDL एक का 35 है, दूसरे का 25 है. तो किसमें CAD का ज्यादा रिस्क होगा? रिमेंबर, टोटल कोलेस्ट्रॉल इज सेम, तो किसमें रिस्क ज्यादा होगा? आई रिपीट दैट वेरी सिंपल क्वेश्चन. अब ये CETP डेफिशिएंसी, LCAT डेफिशिएंसी, इसको आप, ओके, आई टेल समथिंग. CETP डेफिशिएंसी में elevation of cholesterol over fourfold elevation of HDL, oversized HDL, but they have got minimal risk of. Why do they happen? Because, you know, cholesterol ester is not getting transferred to the other lipoprotein. Why? The risk of CAD is minimal. LCAT deficiency, abnormal shaped HDL, because यहाँ cholesterol ester is not getting formed. Cholesterol ester, as we have said, sinks towards the interior to impart the spherical shape. Why there is only a mild tendency to CAD? And what is Tangier disease? Near complete absence of HDL.